preparing to live stream.
Yeah, just we need to mute it there.
Mute us.
Mute the YouTube so that you don't hear yourself. Okay.
Just hold on. For those of you who are around, we're just getting the YouTube thing happening now.
Yeah, so you mute this one and now it's going.
All right, very good. Excellent. So for those of you who don't know, the recording is being live streamed and recorded. So I will share my screen now and start the presentation, a quick introductory presentation for everybody. So welcome everybody to the online Ecopath seminar series, which is obviously being brought to you by the Ecopath Research and Development Consortium. Thank you very much for all of you that are attending and for the people that are in the room, which is great to have actual people in the room. So just some housekeeping rules for those of you who are online and people in the room. For people that are online, please make sure that your name is clear when you entered it into the Zoom settings and when you ask a question. Try and use question in the Q&A for your questions so that we can keep track of it, so that I can ask it to Kieran afterwards. And try and put your organization and country in, because that helps when we do that. If you have any technical questions, then please put it in the chat and I'll have a look at that as well. And note that the webinar is being recorded and it will be available on the EMB YouTube channel and on the Ecopath YouTube channel eventually. For now, it'll just be on EMB because we are not able to go into the Ecopath YouTube channel from this side. So yes, we're hosting it at the Marine Board just because it's easier for me to do that way. I'm a little bit technically, what's the word I'm looking for? I don't really do it very well. So the series is basically held every 2 months on the last Tuesday of the month at 12 noon Eastern time, so Eastern US time. I think it's 9 a.m. in Vancouver, if I'm not mistaken, 6 p.m. in Brussels and 7 p.m. in Thessaloniki, which is where we are at the moment. So today's seminar, and then we have two, it's, as I said, every last Tuesday of the month. So the 30th of November and the 25th of January will be the next one. So today we have a speaker, Dr. Kieran Tierney. He is a researcher at the Department of Civil and Environmental Engineering at the University of Strathclyde. Kieran did his PhD with me a while ago now, and his PhD was on the fate of radionuclide discharge that were discharged by the Cellophile nuclear fuel reprocessing facility. And he used Ecotracer, which is a program, a routine that is part of the Ecopath with Ecosyn suite, which is why he's giving this talk. He's going to tell us about that work, but he's also going to be telling us hopefully about two new projects that he's got just starting using Ecotracer. One is called Modime, which is a modeling-based exploration of the ecological effects of the Wakasio oil spill. And the second one is the R2K project, which is looking at how to predict ecosystem uptake of discharges from the Swedish nuclear facility. So with that, I will stop sharing and Kieran can take it away.
Thanks, Sheila. Oh, it's now not letting me. Oh, here we go. So, hello, everybody. Can you see the presentation? Okay, is that?
Yes,
yeah,
I can. Yeah.
So thank you very much for asking me to present the Ecopath seminar series. Thank you for the introduction. So this is work that I did during my PhD, which was based at the Scottish University's Environmental Research Centre, but I was also affiliated with the Scottish Association for Marine Science and working with Sheila. It's really a story of, if any of you have looked at Ecotracer when you open up Ecopath, why it has this symbol. So Kieran kind of made this symbol during my PhD when I started working with Ecotracer. And also at the same time, Villy and Will Walter started developing some of the code and published a paper at the same time, looking at Fukushima discharges of radioactivity. So this is the Cellophile Ecotracer model. And we published this paper. No. Here we go. So there's a group of us that were involved in this project, and we published a paper on this model 2018, and it brought together a big group of people, lots of different kind of backgrounds, whether it was in ecosystem modeling, also nuclear physics, radiochemistry, geologists, environmental science, and analytical chemists as well, actually. And it was a big kind of project where we were looking at a radionuclide called Radiocarbon. So it's the radioactive isotope of carbon, and it's discharged into the environment from nuclear reprocessing facilities such as Cellophile. So this is a picture of Cellophile. This is what a Cellophile is, and it's located on the northwest coast of the UK, sitting just on the Irish Sea. Cellophile produces radiocarbon just during routine processes, and it is released to the environment, where, because of its high bioavailability, it behaves just like carbon in the environment, and also because of its long half-life, it's got a half-life of almost 6,000 years. It means that it isn't going anywhere. It moves into the biosphere, and it is able to transfer throughout the entire food chain, which means that it's actually the radionuclide that provides the highest collective dose from the entire nuclear industry to human populations around the world. So I'm going to go into a little bit of physics as to what is radiocarbon, but it is relatively simple. And the reason for that is because I wouldn't understand it otherwise. So it is produced naturally. That's how we can do radiocarbon dating. And it's produced in the upper atmosphere, in the stratosphere and troposphere, by cosmic rays. So cosmic rays cause a bombardment of neutrons onto nitrogen atoms. And this causes the absorption of a neutron in the nitrogen atoms, producing C14, kicking out a proton, and whilst it's doing that. Now, C14 is a weak beta decay radionuclide, so that's why it has a very long half-life. What it means is that it actually takes a lot of C14 to be potentially radiologically significant. It's also produced during bomb testing. It was in the 1950s and 60s when there was atmospheric nuclear testing. Not that I'm advocating nuclear testing at Cellophile. And this doubled atmospheric C14 content at the time. But nuclear waste reprocessing is where the C14 I'm going to be talking about comes from. So it's produced and released through atmospheric discharges. But it is also released at Cellophile through pipelines into the Irish Sea as inorganic C14. So it is discharged within the dissolved inorganic carbon pool. And this is particularly important in the last few years because marine discharges of C14 increased in 1994. So the figure here shows the atmospheric and marine discharges from Cellophile annually. And in the 1990s, scrubbers were attached to the flumes at Cellophile to recirculate C14, and it was then released through marine aquatic releases rather than from the atmospheric discharges. Now, once it enters the marine environment, hypothesized that it is going to be uptaken by photosynthesizing organisms because it will behave just as any carbon, any dissolved carbon within the marine environment. And so it's then able to enter the marine food web. And of course, it can also build up within sediments from fallout, and then in calcifying organisms as well during shell formation. The calcifying process was actually a paper that we published some work on that in 2016. Now, mentioned here, there's northward transport of C14 once it enters the Irish Sea, and that's due to the general circulation that exists within this environment. So this is where we are. So a map of most of the UK and Ireland. Cellophile is roughly where the red dot is. And during this project, we sampled a number of sites collecting biota and water. I'm mainly just going to be talking about 3 here. So this is a site in the eastern basin of the Irish Sea, or EB, which is located quite close to where the pipelines come out. One in the West Basin, which is a slightly deeper body of water. Isle of Man sits in the middle. And then the North Channel, which is a deep, narrow body of water that goes up to the west of Scotland. And we had another main sampling site just off the side there. So we'd had water samples, biota samples. Biota included a variety of organisms. And also we had a number of marine mammal samples that we published in another paper, which showed C14 transfer through the entire food web up to, it was harbour porpoises and seals, which are our main kind of resident mammal species that we get around the UK. So the model actually used a repurposed model of the Irish Sea that was published by Lees and Mackinson in 2007. The model was developed from 1973, and that suited us because Cellophile's been discharging radiocarbon into the Irish Sea since the late 1950s, early 1960s. What we did was we edited the model a bit, reduced the number of functional groups just to simplify it slightly. There was a publication, a thesis on incorporating better description of marine mammals within this model as well. And so we incorporated that into this, and following the pre-bulb analytical tests, we made some other relatively minor edits to this model. It wasn't fitted to time series data. We decided that a model that described an environment for that particular time was good enough for the questions that we were trying to answer. This was effectively a feasibility study. Can we use the Ecotracer? Can we use the Ecopath approach in Ecotracer to be able to capture what is happening with C14 in this environment? So this is the food web that we ended up with. So it's still a relatively complex food web, number of functional groups, as well as several species that were described within it as well. I'm not going to try and explain that because you won't really be able to read it. We generated a map for Ecospace that was on a 5 kilometre 5 by 5 kilometre grid. What this meant was that there were some areas in the west of Scotland. So in the bottom here is Irish Sea with the Isle of Man sort of sitting in the middle. We have the North Channel heading up to towards the top left of the map, and then this is into the sort of west of Scotland area. And in here, we lost some definition because of the map resolution. So some of the islands aren't, for example, they aren't well defined here. Some of them are actually joined to land. So this was something that we considered, but we thought, given that the Irish Sea itself was our main focus area, and for the questions we're trying to answer for the west of Scotland in terms of the general extent of C14 transfer and uptake in this sort of more far field area, we thought it would be good enough for what we were trying to do. We got hydrodynamic data from Copernicus, and this is then what we used as for dispersion of C14 within the model area. So we had a point source located just off of where Cellophile is within this map. And it was here that we had our contaminant input into within the Ecotracer. We then used this advection data, which was on a monthly time step, to disperse the C14 inputs into the model. This was depth averaged data. And again, this was something that we considered at the time, but we thought this was probably going to still provide us with a good enough dispersion modeling, good enough prediction power to get the answers that we needed for what it was we were looking at. In terms of Ecotracer input, I'm not sure if you can see this too well, but I'll go through it relatively slowly. Within Ecotracer, your initial box allows you to put in a concentration, an existing concentration of contaminant within the environment. For us, this wasn't important. This wasn't something we need to use because we made up a driver file. So there's a box that lets you load up an Excel sheet and CSV sheet. And with that, you can input contaminant data for a specific grid square or multiple grid squares within your model environment on whatever chosen time steps that you're using, whether that's yearly or in this case, we were doing monthly time steps. You could also do this via a forcing function, depending on what kind of data that you have, whether or not that's something that would be useful to you. There are decay rates that you can add in here as well. Now, we could put a decay rate in for radiocarbon, but it's got a half-life of almost 6,000 years, and we weren't covered in that sort of period length of time, so we didn't need to put that in for us. We decided to go with having effectively a clean environment where no radiocarbon exists, which is obviously not true, as I already told you, it's produced naturally. But in this case, what we're interested in, what was the increased radiocarbon concentration within different species groups as a result of releases from Cellophile. And so for that, we decided to keep all our initial concentrations to 0. Our main or only route for uptake into the food web is absorption or uptake by primary producers. And we based this on actual primary productivity rates for the 3 primary producers in our system. So that was phytoplankton, microflora, and seaweed. Like I said, we didn't include any decay rates because of the period of time that we were considering. Proportion of contaminant excreted and metabolic decay rates, they come from our Ecopath input data. So radiocarbon is going to behave just like any carbon, just like any energy that's flowing through the system. So a proportion of contaminant excreted can be the same as you'd have in your basic inputs. in terms of your excretion rates. And the metabolic decay rate was then, we utilised what Ecopath estimated for respiration rates in your Ecopath output. And we were able to test this, which made C14 quite a good contaminant, as it were, to look at what Ecotracer is capable of doing. So we had a specific environmental concentration that we kept constant, and we had these absorption rates and these metabolic decay rates, and we could see that the concentration in any one functional group, particularly for our primary producers, that they flatlined. So that meant that we knew that our uptake and our removal from groups was equal. So C14 is behaving like carbon. It's not going to bioaccumulate. It's not going to concentrate in any specific group. So I'll show you a video of the actual environmental concentrations within Ecotracer once this has been input in and using the Copernicus hydrodynamic data to force this within the model. So our model started in the 1980s and in 1984-5, and I'll run this again. And even though the model data, or sorry, the actual discharge data for Cellophile chose releases going back into the 60s, we didn't have hydrodynamic data going back that far. So that's what we used as our start point for this. Our main interest was what happened coming into the 2000s when Cellophile discharges were hitting their sort of highest concentrations. And then when we were actually going out and sampling and analysing biota, which were samples that were taken in 2014. So I'll run this again. So we have our releases in the eastern basin of the Irish Sea, and concentrations ranging from 1 into over 10,000 becquerels. So our measurements are all in activities, and they're in activities per kilogram of carbon. We could have used carbon just for our initial masses within Ecopath, but we kept it as biomasses, and instead what we did was we just recalculated our outputs afterwards. You can see that once we had the higher releases from Cellophile, that the activities were increasing in areas in the west of Scotland, up towards the top of the map. But we're already, even at this point, seeing that they were potentially not as high as what we were actually observing in our analysis. What we started looking at was the base dispersal rate within ecospace. So if you look in ecospace for different functional groups, you can set a base dispersal rate for them. And for a contaminant, the base dispersal rate is connected to whatever value you assign for your first group after detritus. So typically one of your primary producers like phytoplankton. What we found was that, so advection is the dominant control on dispersion in our model, but we could use the base dispersal rate to still have an impact on it, acting as a sort of likelihood function for movement of the contaminant to the border and cells. By increasing it to a minimum of 100 kilometers per month, we were able to get the kind of typical dispersion that has been seen in the Irish Sea. We actually had to double it to 200 kilometers a month to sort of see the range in environmental concentrations that had been measured in the eastern Irish Sea during the 90s up until 2000, which is really the only kind of consistent set of time series data that had existed for this area. And it was because we were able to get that sort of range of activities in the model that matched what had been observed, that we decided that was the best sort of model run, that we would use that dispersion rate for going forwards. So going into some actual results based on using these model runs. So we had samples that were collected in June 2014 from the Eastern Basin within the Irish Sea. And the observed data are the black columns on the figure. The sort of darker grey columns is the model data from our sort of best run, this 200 kilometers per month. And so the data here is showing the net C14 activity for different functional groups. So for example, our epifaunal macrobenthos, where we had sampled maybe different brittle stars and starfish, we agglomerated those that data to provide a single value, which we then compared with the model data. And what we found was that the model was given as really good predictions. I think quite typically when you see this data, you may see it on log scales, and we were really comfortable just showing this on a normal scale because the values are so close. It's almost quite surprising. And we weren't fitting to this or anything. We were just fitting to some of the environmental data, like I showed you before, and we were managing to capture these predictions for this particular area. And bear in mind, the model data is just giving you an average value for a 5 by 5 kilometer grid, whereas we're obviously sampling just from one kind of specific point. The other thing that the model showed was this observed trend of increasing activities with higher trophic levels. And when we collected the data, we had to sort of pinch ourselves and look at this a couple of times, because there was obviously, we knew that you would get no accumulation, no bioaccumulation of C14. It should be behaving like normal carbon. However, when we heard back from Cellophile in terms of what they had released at that point in time, we saw that they, when we happened to be out on the boat, they released one of their lowest concentrations of C14 for several years. What we're actually seeing is time-integrated activities that are defined by diet and lifespan of different species. So species at higher trophic levels that are feeding higher up within the system and usually living a bit longer, the C14 activity that they're going to exhibit within their tissues is going to actually be representative of C14 that has been in the environment over a number of months, potentially even years, depending on the tissue that you're sampling and the species that you're looking at. And obviously, when you're looking at it from a modeling perspective and you've got your time steps within a model, it makes sense that a model would replicate that in some way. But we definitely think that the Ecopath EWE approach and the data that's required, the ecological data, the diet, the information and stuff that's going into this, it really helps, particularly when looking at something like C14, which is going to be behaving like normal carbon. We move over to the West Basin, so we're sort of intermediary site, and it gets a bit more mixed, and it's really a result of having really highly variable data that we collected from this site. We're now, I think, also starting to see potential issues with the dispersion within our model and using Ecopath to do our dispersion. I'll talk about that in a little bit later. Again, the general trends of higher activities with higher trophic levels that we observed were replicated with the model. And where there was maybe 2 or 3 functional groups which showed very different observed activities compared to model, there is some ways we can explain this. If considered the Infauna polychaete group, this included a highly predatory species, Aphrodite, which the measurements of that species alone had activities of several hundred becquerels, whereas other smaller polychaetes, their activities were well below 100 becquerels, and actually showed a value that was much more similar to the model. There could also be an element of fish species like dab that are moving around about in the Irish Sea a little bit more, and maybe been feeding in an area that's slightly more active and kind of coming over. So again, it just kind of shows the importance of how species and such are defined within a model. We head up to a far field site in the North Channel. And our model activities are significantly below what was observed. When we go to higher trophic levels, we are in the sort of right ballpark. We are in the same order of magnitude, but at lower trophic levels, that wasn't the case at all. It looks like the way we did the dispersion within the model, it wasn't the best way potentially doing it here. We depth averaged the hydrodynamic data, and where you have areas like the North Channel, which is a particularly deep body of water relative to the area, you do have multiple currents that can be running in this area. And so you may have a high dispersion of C14 going northward, but it may have been getting cancelled out a bit in the model with currents, residual currents that are moving southwards from the Atlantic Ocean. And so we may have been caught out a little bit by attempting to model in this way, and could have been better using a 3D physical transport model instead. Just another little video. This time show the differences between a species at a lower trophic level, so zooplankton, with one of our species at higher trophic level, harbour porpoises. And I'm just trying to highlight this idea of the time integrated activities that we're capturing. So on the side that has our zooplankton functional group, we can see that the C14 activities are changing a lot. They're very highly variable as the environmental concentrations are varying quite quickly. But that isn't the case with harbour porpoises. Now, there have been sites in the past that people have studied C14, and conclusions have been made at some of these sites that it is reasonable to use environmental concentrations, and by using environmental concentration, you can then get an estimation for what you would, what you think the activities should be in some species. What we found both as in our observations when we're out doing the sampling work, and then what we're capturing in the model, is that in a highly with a contaminant that's highly varying in its concentration, and well, particularly specifically to C14 in this case, that's quite clearly not the case. Things like the longevity, the lifespan of the species in question and its data are going to be so important with regards to what its activity could be at a point in time. You could see very low environmental activities, whereas you have an organism like a fish or a marine mammal within that environment, which is exhibiting quite a high activity. The figure on the left here is just model data really kind of illustrating that point, where you have this really highly variable activities and dissolved inorganic carbon. So this is our environmental data that we're pulling out of the model, and zooplankton, which is sort of following it relatively closely at these different sites. But then when you move through the food web up to organisms at higher trophic levels, we have a much more kind of smoothed out activities for these organisms. This is something that we said in a publication we made on the data that we collected for the west of Scotland and for the North Channel, where we saw some higher activities there compared to some parts in the Irish Sea, given that we expected that there must have been a spike of activities in the west of Scotland when there had been a spike in discharges from Cellophile, which may have resulted in higher activities being incorporated into tissues of some organisms, which were then not apparent in organisms towards the top, towards the bottom of the food chain, like zooplankton. So just to go into some conclusions from what we found by using Ecotracer to try and model this system. So we do think that it is a tool that can be used to provide reasonable predictions for C14 uptake and transfer through the food web. And they're significantly assisted by ecological data utilised by the EWE approach. I think I kept going back to this time integrated activity, which you can't capture through other modelling means as easily as seems to be apparent for using the Ecopath approach. And the other nice thing is there are so many Ecopath models for various environments out there, and they can be redeveloped and used for contaminant tracing, whether that's routine discharges like we were looking at here, or whether there's an incident that occurs in one particular area. It's really important to consider how you approach modelling the contaminant dispersion. So we showed here that for the immediate area from where the releases were being made at Cellophile, we did appear to capture what was happening with C14 really well. But as we moved further and further away, the approach we took was probably not the best approach. And if we had maybe used like a 3D physical transport model, and that may have meant that we would have got better quality predictions for other areas. And then obviously you need to think about your species in your Ecopath model, going right back to your basic input. You're not going to get accurate data out for a functional group that describes species with very different ecologies, like we had with our polychaetes. I'm continuing to work in Ecotracer, so I'm currently at the University of Strathclyde, but I'm moving back to where I did my PhD in November to the Scottish University's Environmental Research Centre, and I'm working on two different projects. One of these is a sort of very similar to what I did for Cellophile, and this is looking at C14 discharges from a Swedish nuclear power plant into the Kattegat, so between the North Sea and the Baltic Sea, but also looking at other contaminants. So, for example, oil. And last year, you may have heard that there was an oil spill off the coast of Mauritius when the MV Wakashio hit a reef. And so I'm involved in a sort of larger group looking at the dispersion of that oil and the effects that it may have had. And we're going to try and bring that into an Ecopath, into an Ecotracer model off that area as well. So thank you. And if you have any questions, and if any of that, I'm quite happy to answer them.
Thank you very much, Kieran. Natalia has raised her hand, so you get to go first, Natalia. You can put your video on if you want. Maybe you've done that already. And
I was thinking to raise the hands, and I done it.
Sorry, Natalia. Yeah, Kieran, you've stopped sharing your screen. Great, thanks.
Thank you, Kieran. I was nice. I heard that background of this presentation many times, and every time I have pages of notes. So especially now, then I am trying to understand it more deeply because I'm planning to use it. I do have several questions. One is right at the beginning, and there's a technical question. you said then the absorption rates, you change just for phytoplankton.
Yeah.
And that is the equivalent of a turnover rates, how you get that? You said it, but I just missed it.
So for C14, the carbons in C14 is going to behave the exact same as any stable form of carbon.
Yeah.
And so as a result of that, we know that the uptake rate of it is going to be the equivalent of the photosynthesizing organism. So at the rate that, for example, your phytoplankton are photosynthesizing, then whatever concentration of C14 is available within the DIC pool, that is how much they are going to then uptake. So for C14, going to be particularly specific for that, and actually makes it quite easy to estimate an uptake rate for. So it's quite a good contaminant in that case for doing this.
You use a normal uptake rate of carbon.
Yes.
And then I do have other questions. I really like I think that's quite important. The fact that you add observations, then you use for validation, so you can set the dispersion rates to try to figure out how much is the dispersion rate to validate what is in the environment. And that is very important. You said then there is not accumulation for this kind of radiocarbon, but also then the radioactivity was increasing with trophic level. So somehow there is a kind of accumulation. I don't know if accumulation is the right word.
Yeah, no, that's right. So, so it's what I was saying was we're getting a time integrated activity for some organisms. And it's just basically based on their turnover rates and their diets. Those are kind of the key kind of components there. So we're measuring the C14 concentration within the muscle tissue, and they're only going to be releasing C14 depending on, so there'll be respiration, but then also the degradation or turnover of cellular mass that's happening in those tissues. But if you have, so there'll always be carbon that's turning over within your own system. So there'll be a remnants of things that you ate several months before still within your tissues somewhere significantly later. So that was why we were seeing for organisms further up the food chain that their activities could be even orders of magnitude above the environmental concentration at that point in time. We got a nice paper out based on a marine mammal data on that. I think it was the 2017 paper, and we were able to show that the activities in a stranded harbour porpoise on the UK coastline were significantly linked to what cellophile discharges had been 2 years prior. And what was nice was that within the Irish Sea, we could say that the activity in that animal was linked to what the releases from Cellophile were between 0 and 24 months prior to when it died. But if it was an animal that stranded on the west coast of Scotland, it was between 12 and 36 months before. Okay. And even though we knew that there was genetic mixing between the west of Scotland porpoises and the Irish Sea porpoises, we were then able to say that, so you may have some boys moving up and down and mixing, they're not feeding in the areas that they're maybe moving into. They've probably got really high foraging fidelity for, you know, whether they're Irish Sea animals or west of Scotland animals.
Thank you. And I do have a last question.
No, Villy's got his hand up. You can't have all the time. Go, Villy.
I'm actually wanted to do this just to demonstrate that I can get Natalia to stop talking. And we all know how difficult that is. Thank you very much, Kieran. It was great to hear about the projects which I knew about and the 2 new ones that I look forward to hearing much more about. For this kind of modeling, there's an issue that the water in the models where that is modeled moves with time unit of seconds, and we're using months in the standard Ecospace model, the ones with time series. And that connection there between the hydrodynamic models and the ecosystem models is an area that really needs a lot of consideration, and we need to work with good hydrodynamic models. I think that's quite clear. Lots of things have happened there. What's your experience of the, you've been using output from monthly output, average monthly outputs from models. So what kind of issues did you encounter there? I think we saw some with the North Channel more clearly than for a few others, but what's your experience here?
My general experience was, my main thought or issue wasn't necessarily so much using month average, because I think you're still going to capture the kind of general current system that will exist in large bodies of water. I think if you were focused, so particularly I guess on the west of Scotland with a lot of the sea locks and narrow channels, there will be effects in there that are not going to be getting captured. But I think that the main issue was potentially the depth averaging of data. And where we have a body of water like the East Basin within the Irish Sea, which has a depth of an average of around about 30 meters, and there's not really anywhere with any kind of significant depth. But then you hit an area just north of it in the North Channel, and that's the kind of main route for contaminants that are in the Irish Sea for moving out of it, for their main kind of dispersion route. And you have a really kind of mixed current regime within that kind of deep, narrow strait, with currents that are moving at different directions at higher and lower areas. It's, you're not going to capture them when you're depth averaging data, especially I guess on these kind of longer time steps. And at worst, what you might be doing is just sort of flatlining your advection. So even though we still got a general northward movement, it probably wasn't strong enough within our model to kind of capture what the reality was beyond the Irish Sea.
Yeah, we've seen something of that here for modeling Strait of Georgia, where Greg Oldford has developed a hydrodynamic model and the ecosystem model, and it's showing actually once you have time model, we'll find times that you can get a lot of dynamics around fine structures, islands, small openings, and so on. That is really important to capture for the other model. So this whole, I think it's really important that we have this cooperation between the hydrodynamic models, and that they to provide the water flow basically with all. How we do that and how we get, in your case, how we get the dispersal right, I think is really one of the things that we need to work much more with. Yeah, but it's great to see this cooperation.
I'll just add, is one of the approaches we're going to try and take with the Mauritius project. So we're linked up with NOAA, and we're going to try and do the actual oil dispersion from the ship based on one of the NOAA models. And then we'll feed that into an Ecopath model using the spatial temporal framework, rather than using the framework for our advection data and then feeding the contaminant in from a point source.
For some other work here that I was involved in, which had to do with smolt coming out of the Fraser River and dispersing there, where we're looking at the fine scale, I simply ran Ecospace with daily time steps, so 2-hour time steps. Actually, no, in the end, I went down to 12-hour time steps. And that means which means every hour a new field, because the tidal cycle was really important for that. And I could imagine for oil spills where you're talking about a run length of that's in days or weeks. I ran for a month for that model. I think it's worth considering.
Yeah.
Changing the unit, and if you make it 12-hour time steps, it fits in. You can read for every hour. You can read in a new field, and I think much of the fine dispersal would be captured much better in such by such a setup. It was straightforward to set this up. I was only using tidal currents, so it was fairly simple in this case. Here, I'm responding to the question from Christy, and no, it wasn't. It was just a few days' work to set this up and to get it running.
A few days work for Willy, which is like 5 months for us, right?
I think the main thing there was what took time was rather to figure out what to do to end up with that hourly time step. And you know, this ended up being quite a nice evaluation of if this was impact of a terminal on water currents and so on, and mainly showing that this area we were looking at was dry half the time because the tides and how that influenced the mixing of the small. But setting it up was pretty straightforward.
Thank you. I don't know. I think Christy, I don't know if you have another question. I think we've made you co-host now, so you should be able to show your video. Oh, okay, maybe we haven't done that yet.
Yeah, I can share my.
But Natalia, do you want to go with your other questions if you have it?
Yes, thank you. I was scared to raise my hands now. A simple question, really. You said that we can expect to have good output for functional groups then they are very diverse, like polychaetes. If you could back in past and do it all over again, would you simplify the model even more, or would you do the opposite, just have more functional group to better represent them?
There would be a combination of both. I think there would be some things that would be simplified more and other things that we would maybe try and look at pulling out more detail. I think if I was going to do it again, I would try and convince all the analytical chemists that I was working with that actually I only need to spend a year with them and I need to spend 2 or 3 years on actually the modeling rather than the other way around. But the nice thing was I was only able to show some of the data there that we were then using to look at the output from the model. But I think we generated more data on this particular thing on C14 in our environment than has been generated before. And it is going to be a particularly important thing for a lot of areas. So obviously I'm now going to be working with Swedish Radiation Authority because they're now interested in what is happening with their C14 in the environment. And it's because all kind of nuclear nations, they have a huge inventory of radiocarbon. The UK has over 200,000 tons of graphite bearing really high levels of C14, which we need to be able to sort of safely lock away for thousands of years. And because of different regulations, different policies, some of them maybe Sheila will be aware of as well, but it means that these nations are being told that none of that can ever get into the marine environment. It doesn't matter whether you can say it's not going to cause any real damage or not. There's still a lot of unknowns, within radiochemistry, so people are not willing to take any of these risks.
Thank you.
Thanks, Kieran. So there's a question from Tofu Zeglo. He has a question on the concentration of radiocarbon. Is there a difference between pelagic fish and benthic fish in this context? And is there an impact on migratory fish?
That's actually that's quite a nice question. Well, nice. It's not easy. We found that there was a difference in that we saw increases in the activities within pelagic fish before demersal fish. So if there was a spike in releases from Cellophile, then pelagic fish would their activities were going to increase first, because I guess they're closer to feeding on zooplankton which are feeding on phytoplankton compared to demersal fish. Some of our demersal fish species, when we're measuring the west of Scotland, also quite typically actually had some really low activities. Like we had samples of monkfish, which I think registered almost near background, whereas there was maybe species like whiting, which had activities that were several hundred becquerels above what was background. So there's there's lots of different things probably going on there. Migratory fish would be interesting. So we made a point of not looking at other marine mammals because we wanted to see what were the activities of species which were resident, which were always going to be in that area. In terms of migratory fish, I think it would be a similar thing where they may pick up a little bit of C14 as they're passing through, and that may remain within their system for a little while, but then when they're feeding elsewhere, that's going to be lost relatively quickly. We did start looking at otoliths from cod and haddock from the Celtic Sea to try and see if there was any kind of we could see any migration from the Irish Sea into other fish stocks for these species, and it was rather kind of inconclusive. The activities just maybe weren't high enough to be able to kind of show that.
Thank you very much, Kieran. I don't know if there's any other questions, but I think we're kind of at the end of our hour. So any other questions from anybody? If not, I want to thank you all. Thank you very much, Kieran. That was great. It's good to hear about the other projects, and it will be interesting to hear how you get to actually model the circulation or the dispersal of oil, and how you get to bring that into the ecosystem. I'm quite interested to see how you get that into the food web. It would be interesting to see. So if there isn't anything else, I will remind you that the next webinar will be in November. I've forgotten the date now. And we will let you know again through Facebook and so forth of when you can register for that. And this time I'll try to do it quicker. Vili, you look like you want to say something, one last word. We can't hear you. No? No? All right, so if there's nothing else, then I just want to thank Kieran and thank everybody else who came online. And yes, we will keep this on the YouTube channel, both Ecopath and Marine Board. So if anybody's interested, you can see it there. So thank you very much.
Thank you.
Thank you.
Bye.
Bye.
Well done, Kieran. It was really great. I really enjoyed it.
Thanks, Lee.
Thanks, Kieran.
Thanks, Jacob.
I couldn't put my camera on for some reason, but it was really good, man.
Cheers, man.
Nice to see and hear you again.
No, thank you. Thanks. Hope you're
Good luck with
doing
those
right.
projects. They sound really exciting.
Yeah, like kind of just really started. Not even started the Swedish one. They're kind of going to be happening.
Understandable or a little bit? It was a bit kinky.
Oh, nice. Well, I look forward to seeing what happens. Cool. All right, I'll speak to you in a bit.
Take care, Jacob.
Bye.
Bye.
Bye, Sheila.
Bye.
Thank you for hosting.
Thanks, Sheila. Bye.